Introduction. Good morning or good afternoon, ladies and gentlemen, for this webinar. Today, I would like to talk about water analysis using analytical protocols and instrumentation which were developed by Lumex Instruments Company. For this analysis, we offer to use capillary electrophoresis method, and we'll talk today about how it can solve many analytical tasks in the water treatment industry. This is actually what is the title of the presentation: Fast, Accurate, and Reliable Chemical Analysis of Water. We will talk at the beginning short. I will introduce you our company, Lumex Instruments. Then I would like to analyze some basic features of capillary electrophoresis as a method, which was realized on our CE instrument. CE is actually the abbreviation for capillary electrophoresis. Then we'll talk about the task in water treatment industry, about the specific task. I will underline what is the specific task in this industry. And finally, we will discuss in all details the most important analytical protocols to be used for water analysis, which we also developed in Lumex Analytical Center and realized on our CE instruments. But let me first introduce our company. Lumex Instruments is worldwide known as a company which manufactures analytical instruments and develops the necessary analytical protocols to be realized on them. The field of application of our instruments is broad, from the life sciences to pharmaceutical, academical research, food testing, and so on and so on. Our headquarters are located in Vancouver, Canada, and in St. Petersburg, Russia. We have important sales and research offices in Hamburg, Germany, and in Beijing, China. More than 350 employees worldwide are working in Lumex, and we export our product in more than 80 countries. On this map, actually, you see everything which is colored in blue are the countries to which we export our products, and we are really proud of this fact. On this slide, you can see the most important and well-known instruments from our company manufactured by Lumex: miracle analyzers, fluorimeters, atomic atometer, infrared analyzers, PCR analyzer, of course. And today we are going to talk about the capillary electrophoresis systems named Capel, and the first Capel instrument was sold already in 1997. Of course, the instruments which we have now on the market, which we offer to the market, looks a little bit different than it was before. Here you see a short list of fields where these instruments are actively used, from pharmacological industry to biotech research, including peptides, proteins, immune analysis, but also in food industry, and of course the environmental analysis. There is a very important role in the field, between the field where our Capel CE systems are actively used. To understand better what we're talking about, let me now shortly explain how actually the CE method works. The heart of any CE system is a narrow fused silica capillary filled with an appropriate buffer called the electrolyte. And this is very important to underline. This capillary is filled just with the electrolyte. There is no any sorbent or solid material inside, in contrary to any kind of liquid chromatography. Capillary is being thermostated, and there is an inlet and the outlet spaces in the instrument. The vials contain the same electrolyte which is inside the capillary, and there is a high voltage connection between vials. In the outlet part of the capillary, there is a so-called detector window, and it is connected with the computer. So at the beginning, we fill the capillary with the appropriate electrolyte buffer. It could be the very standard borate buffer or phosphate buffer, nothing special. After this, we put the sample solution under the inlet part of the capillary, and using the very precisely controlled, accurately controlled injection, we inject a tiny amount of the sample solution in the inlet of the capillary. We are talking about 2 to 10 nanoliters of sample solution, not more. After the sample solution is injected, we put back the buffer vial under the capillary, And the sample vial can be taken away, or you can use the sample vial, or you can use the sample solution for any other purposes. And right after this, the high voltage is being applied. Components are charged. Components of the mixture, of the studied mixtures, which we injected in the capillary, are charged, and therefore they start moving along the capillary. But they are differently charged, and therefore they move with different velocities. Therefore, at the end of the analysis, we get the sequence of peaks at the detector point, where peak area and peak height is directly proportional to the initial concentration of these components in the studied sample solution. Thus, we have the classical separation technique like in chromatography. But in contrary to chromatography, as I mentioned already several times, here capillary is filled just with a liquid buffer, no any solvents inside. Here, the separation is due to the differences in electrophoretic mobility. This is very shortly how any capillary electrophoresis instrument works. One of the most important features of CE is a so-called electrosmotic flow. I will not go deeply into details explaining how it works. Generally, it is just a bulk flow of the whole liquid inside the capillary when the high voltage is on. This gives rise to the significant difference in flow profiles between, for example, HPLC or any kind of liquid chromatography and CE. In case of HPLC, the profile is a parabolic shape because we use pump to provide pressure to press the liquid phase through the column. In case of capillary electrophoresis, the profile is flat because we do not have any pumps and there is an electrosmotic flow which drives the liquid through the capillary. It's understandable that this leads to a much higher separation efficiency in case of CE. So let me now summarize the most important features of CE. First of all, once again, it's very important to understand separation occurs under the influence of the applied electric field. There is electrosmotic flow, which results in the absence of any pumps, and it gives a very high peak efficiency. We don't need solvents inside the capillary. The capillary is filled with just a buffer with electrolyte. It means that we do not have such known chromatography problems like unspecific binding with the solvent, no problems of aging of the solvent, no problems of shifting the solvents, which drastically shifts migration times, and so on and so on. And finally, we deal now with the separation technique. It means that we get the multiple information about the multiple components is just in one run. The next slide is very important to understand our marketing strategy. There are actually two different groups of potential customers to which we offer our products. First group is scientists working in different research centers. For them, we offer our sophisticated CE systems from Capel series in which CE method is realized. They can use the instrument for scientific investigations and method developments. They can publish results in different scientific journals or present them on symposiums on this topic because the CE method has been a subject of science since more than 35 years now. We are always joking that even if the instrument will be completely damaged, you can still use it as a thermometer. It measures temperature. But seriously speaking, this is a very sophisticated instrument which we are proud to have on the market. Totally another group of potential customers is analysts working routinely every day with different types of analysis in analytical labs throughout the globe. They need not just an instrument, but the complete solution of their analytical task. For such customers, we offer the very well-developed analytical protocols, or better said, protocols, analytical methods, or better said, let's say, protocols for the determination of this or that group of components, which are realized on Capel CE system. These protocols are composed in the form of SOPs, or standard operation protocols, which means they can be internally or externally validated in each accredited lab according to the very famous normative document ICO 17025. And actually, this has happened already in the labs of many of our customers worldwide. Most of our protocols were internally and then externally validated and certified in many accredited labs. As I mentioned, we have two things to offer. We offer two parts. First, the instrument. Let us talk about the instrument. This is on the photo. You see one of the latest modification of Capel instruments, Capel 105M. We have a spectrophotometer detector with spectral scanning mode to facilitate the peak identification. We have definitely one of the best sensitivity on the market, down to low ppb due to patented optical scheme. We have capillary thermostating with water to ensure highest reproducibility. Liquid thermostating is always more effective than air thermostating. And besides, we use, as a coolant, we use just distilled water. It's no any special liquid used, so it costs really nothing. We have special high pressure mode to work with viscous buffers. This could be very useful when you deal with capillary gel electrophoresis to analyze protein, for example. And we have independent carousels in the outer sampler for 10 and 10 positions in case of previous modification of Capel 105M, and for 59 positions on the very, very last, the latest modification of Capel. So it's the photo of which you see on the bottom to the right. Actually, water analysis by CE, how it is regulated. There is international standard from ASTM or EPA. You see the numbers on the screen. It tells us how to determine inorganic anions in water samples. In all details, it is stated there. But besides, Lumex-developed several national and international standards for the water analysis by CE. And these are determination of inorganic anions and inorganic cations in water samples, and also some other protocols which we will talk today about. Let me underline, this is important, that both standards which you see on this screen can be realized on Capel instruments, which means that it's a matter of choice for our customers to decide which protocol to be used with our instrument, the ASTM EPA or the protocol coming from Lumex Analytical Center. Water analysis. So where actually it should be used? It's a huge application field. First of all, it's a monitoring of surface water. Getting knowledge about what is with the surface water, we can understand much better the processes which are occurring in nature. Manufacturing of potable water, actually all 7 billion people living on Earth need every day a huge amount of water. So you might understand what a huge industry is this manufacturing of potable water. Mineral water analysis. Not each mineral water can be consumed because it might be dangerous for the health, and therefore it must be measured again and again. Content of it must be measured. Water treatment stations. A huge amount of water is being treated, wastewater being treated every day. It must be measured and analyzed at the beginning, during the process of treatment, and after the process of treatment. Water preparation for other industries. It is very important. For example, if we take the beverages industry, production of beer, you understand that the major component of beer is water. And if you produce beer, you must analyze water many times during the technology. Analysis of various water extracts. Capillary electrophoresis can analyze only aqueous or aqueous organic solvents. But if you have your material to be analyzed in a solid state, you can make the water extract and then analyze the water extract by CE. So this is indeed very important. We now talked about what was developed by Lumex. Lumex offered to you several very well developed protocols: determination of inorganic anions, determination of inorganic cations, determination of organic acids, determination of products of water chlorination. And several other groups of compounds as well can be measured and determined, but this is the list of the most famous and the most well-known protocols from Lumex. Let me now pay a special attention to the specific tasks in water treatment industry. We talked only about the general tasks, but what are the specific tasks? It's quite a lot. For example, how to regulate the efficiency of water purification by membrane technology. You know that membrane technology now is one of the most popular and actively used for water treatment in developed countries. But it might be very dangerous because membrane can cut almost everything which is inside the water. And at the end, you could get just a distilled water. And distilled water is dangerous water if you consume it in a big quantities. In a big quantities. So you must analyze water before the membrane and after the membrane. This is one of the specific tasks. The next, how to keep alive microorganisms in a wastewater treatment station. Microorganisms play a very important role in the wastewater treatment stations. They consume the organic substances in the wastewater and convert it into non-hazardous compounds like CO2 or NO2 or just water. But these microorganisms can rapidly die if there is something absent in the water passing through this biogel, or in contrary, if something is in a big excess. For example, phosphate. Phosphate is absolutely necessary to keep these microorganisms alive. But if phosphate amount will be higher than a certain limit, microorganisms will die. The same situation is with sulfate and nitrate. So it is very important to control the presence of inorganic anions in wastewater when you pass this water through the biogel. Desalination, when to stop. Many countries use desalination as a method of choice to get the potable water, especially countries which are located on the very hot part of the planet and on the sea or ocean coasts. So they have a huge amount of salt water from ocean, and it must be desalinated. But when to stop? Because the desalination normally is the distillation process. And at the end, you get a distilled water. Once again, you should not use the distilled water for a standard household purposes. So you must stop before when there are still some salts dissolved in water. When to stop? This is the question which can be answered by regular routine measurements done by capillary electrophoresis. Manufacture of bottled water. Minerals out, minerals in. When you manufacture the bottled water, in most of cases, first of all, you take all minerals out from the water which you use as a source, for example, in natural or surface water. But after you do this, after you have done this, you need to add certain minerals back again, because once again, you should not use the distilled or semi-distilled water for the household, for the consumption. Therefore, you do several processes, and you can save time and money by measurements of water, or the salts dissolved in water before, during, and after this treatment. And of course, we have many other tasks as well. The detection which we use in our system to detect inorganic anions or cations is called so-called indirect detection, because cations like calcium, magnesium, or anions like sulfate, nitrate, nitrite, and so on, they do not absorb in ultraviolet light. In order to make them visible, we add a certain chromophore in the buffer. And thus, when zones are being formed, zones of separated solutes, in these zones we have the negative peaks. Just mathematically, we invert these peaks into positive ones just in order to make them, let's say, more user-friendly. This is our indirect detection what we use. Let me now show you for the first time how it looks like, the results of the analysis. I mentioned before that we have the international regulation document coming from ASTM and EPA. So this is how it is analyzed. It is chloride, sulfate, nitrate, nitrate, fluoride, and phosphate. On the top trace, you see the analysis of the control mix, and on the bottom trace, you see the real analysis of the tap water where chloride, sulfate, and nitrate were found and quantified. This is EPA STMA standard. But Lumex, as I mentioned already, has developed their own standards for inorganic anions and inorganic cations. On the left part of the slide, you see the typical analysis of control mix, let's say calibration mix of inorganic anions, bromide, chloride, nitrate, sulfate, nitrate, fluoride, and phosphate. And on the right side of the slide, you see the typical analysis of the calibration mixture of cations. It's ammonium, potassium, sodium, lithium, magnesium, strontium, barium, and calcium. Let me also stress your attention that with the help of the protocol for the determination of cations, you can see also manganese, and you can see also some important amides, but we will talk about this later. Now it is important to underline that Lumex provides detailed protocols and kits for both analyses in order to simplify the life of any analyst. You might understand that we in Lumex, we have thousands of examples of analysis coming from our customers all over the world. Some of them I will show you here. This is a very typical analysis of tap water. Here it's on the top trace, it's from Switzerland. On the bottom trace, it's one of the mineral water. We have standard situation with chloride, sulfate, sometimes the traces of nitrate. Sometimes nitrate is present in a big quantity. We have traces of fluoride and sometimes traces of phosphate. All analysis being done in 4 minutes, 5 minutes, plus 2 minutes to condition the capillary, which is really nothing. So one sample can be analyzed within 6 minutes. Here you see other examples of how our analytical protocol for the analysis for the determination of inorganic anions and water samples can be used. Wastewater can be analyzed. What you need is just filtrated from membrane, filter, and centrifuge. That's all. Groundwater from Japan, you see it on the bottom trace. No any special things. Just let me stress your attention on the top trace, wastewater. We see the traces of nitrate, which normally do not present in the standard water samples, but this is the wastewater. Therefore, we have this nitrate. And let me also underline what an excellent sensitivity is of this method. The nitrate is being detected on the level of 200 ppb, so 0.2 milligram per liter. So it is one of the best sensitivity. Fluoride is being detected on the level of 100 ppb. So it's excellent, really excellent sensitivity of the method and the sensitivity of the instrument which we use. Let me now stress the main advantages of capillary electrophoresis. First of all, it's a high flexibility in method development. So many applications can be realized on one instrument. What does it mean? I will show you later the example. Electrolyte is a buffer. The electrolyte, which is used for the separation and which is inside the capillary, it is just a buffer. Buffer which has certain additives, some components, borate with SDS, for example. By varying the concentration of one of the additives just slightly, you can get the separation of the peaks which were not resolved in the previous formulation of your buffer. This is the reason why we have a high flexibility in method development. We do not need to use thousands of factors which might affect the separation of our components. What we need is to play around with pH. Sometimes it's enough. And with the concentration of the main additive to the buffer to get another analytical protocol. The second point, easiness in switching between different protocols. We call it universality, versatility. How it's done. So we have a capillary which is packed in a capillary cassette. When you wish to switch from one protocol to another, you withdraw the capillary cassette from the instrument and you insert inside another capillary cassette, which was reserved for this certain application. It takes you several seconds. So one instrument can be used before noon for the analysis, for example, for the determination of inorganic anions. Afternoon, it can be used for the determination of inorganic cations or organic acids or whatever. It's very easy to use one instrument for many, many analytical protocols. Simplicity in instrument design is very important because we don't have columns, we don't have pumps like in HPLC, we don't have a high pressure, and consequently we don't have the special connections which must sustain the high pressure. Therefore, it is definitely low initial price of the instrument. And finally, we have very low reagent consumption. It's just up to 6. Sometimes if you work extremely intensive, maximum 10 milliliters per day. It's nothing compared with the weights which you have normally from any kind of liquid chromatography if you work routinely all day with it. These 6 milliliters per day of the solutions, or weight solutions, it's just buffer, borate or phosphate buffer, which you can consume in a standard way just by throwing them away. You cannot do it with the HPLC waste because you have normally liters of mixtures of acetonitrile with, for example, phosphoric acid, and these liters cannot be consumed in the standard way. Another protocol which we're proud of to have on the market, to offer to the market, is the determination of inorganic cations in water samples. Once again, I mentioned that we can find ammonium, potassium, sodium, magnesium, lithium, strontium, barium, and calcium. Here on the top trace, you see some wastewater from military zone. We have a lot of components. Mineral water from mountain region is on the bottom trace. Here on the next slide, on the bottom trace, we have wastewater from industrial region, and it's surprising. It's almost a half of the Mendeleev's periodic table of elements, which you have which we have here, including strontium, and also it's a peak, which comes right after barium. It is manganese. I mentioned at the beginning that manganese also can be easily determined with this protocol. Analysis of water extract. You might remember in one of the first slides I mentioned that water extracts can be also analyzed by CE. In this case, it's a corrosion study. On the left part of the slide, you see the photo of the metal part with a very visible traces of corrosion. These traces are taken from the metal surface and incubated in water for a certain time, by shaking, for example. And then this water solution, this extract, is filtrated, centrifuged, and directly injected in capillary electrophoresis instrument. That's all. And on the right side of the slide, you see the results. And here, this brings me to the very important point I mentioned at the beginning. We have the highest flexibility in method development. We changed slightly the concentration of one of the additives to the inorganic anions buffer, and now we are able to separate inorganic anions from organic acids. Thus, we get the information about all 6 most important inorganic anions: chloride, sulfate, nitrate, phosphate, but also simultaneously in one run, we get the information about the oxalic acid, about the formic acid, and about the acetic acid. And this, in this certain case for the corrosion study, it's extremely important to know what was the cause for the corrosion. We can answer this question by getting the information about what organic acids and what inorganic anions are present in the traces of corrosion. Next, analysis of water extracts, the studying of soil, very important in agricultural sector. Here we use soil, we mix it in water, we extract it by shaking, then we filtrate it, centrifuge, and directly inject in a capillary. Here you get the information about all these 6 important inorganic anions, and also, as I mentioned, about 3 important organic acids: formate, acetate, and oxalate. Of course, each peak, each component is being precisely quantified based on the calibration. Chlorination product. The chlorination is, unfortunately, I would say, it's still one of the most popular methods to treat the water. Hazardous components can come out of the chlorination procedure. These are chlorate, perchlorate, and chloride. We in Lumex, we developed the method how to analyze them with a very, very high sensitivity in tap waters. Here you see 2 samples on this slide, 2 different water samples after chlorination with sodium hypochlorite. In each case, we were able to visualize and to quantify all 3 hazardous components: chlorate, perchlorate, and chloride. On this slide, you see the very rough estimation comparison of the expenses and cost between CE instruments and the very standard ion chromatography system. You see, in each step, starting from the initial investment, CE is favorable. But especially it is favorable in case of waste. I mentioned already once that the regional consumption is extremely small in case of CE. If you work 21 working days per month, you get at the end about 130 milliliters per month, which is nothing, and this is not hazardous solutions, it's just standard buffers like borate or phosphate buffer. This is uncomparable with waste for standard ion chromatography when you have about 23, 25 liters, sometimes 30 liters of hazardous waste solutions, which you cannot consume in a standard way. So this slide, this comparison shows how favorable CE can be in contrary to HPLC, to any kind of liquid chromatography. On this slide, let me summarize the very important economic considerations why it is better to make the choice in favor of capillary electrophoresis. Very low sample consumption. As I mentioned, what we inject in a capillary is about 10 nanoliters of solution. Of course, if you work with studying of the natural water, any kind of water, tap water, bottled water, and so on, it's not a question for you. It's not an issue for you. But if you work with some extraction and water extracts, if you work with the biotech water samples, it is definitely a very critical point for you. Minimum sample volume, which must be in the vial to be injected, it is very low. It is normally about 20 microliters, not more. We have very low region consumption, as I mentioned, not more than 10 milliliters per day. Standard is 3-6 milliliters per day. And all these points bring us to the very low analysis cost because we have very low initial price, we have very low region consumption, we have very cheap consumables because the capillary, which is the heart of the CE system, costs almost nothing compared with the column in ion chromatography or in any other kind of liquid chromatography. And all these things together bring us to the very low analysis cost. So let me really hope that it was quite interesting for you, and the combination, this unique combination of instrumentation which we offer to the market and the certified analytical protocols to be used in water analysis. This combination could be the very right choice for you. So thank you very much for your attention.
Well, thank you, Vadim, for that very interesting presentation. Now, we do have some questions from our audience members, which we'll take a look at in just a moment. But first, let me remind our audience that any questions we do not have time to answer today will be answered via email after this event has ended. And don't forget, this event will also be on our website within 24 hours' time for on-demand review. So let's take a look at the questions. Our first question, Vadim. Is using a spectrophotometer to check for bromate in drinking water as accurate as ion chromatography?
Absolutely. We can measure bromate in drinking water without any problems, and we can use the same system which is established already to analyze, to measure other inorganic anions. Yeah, no problems.
Thank you very much, Vadim. Our next question. How is it possible to use so many analytical protocols in one instrument? Can you explain in slightly more detail?
Yeah, of course. So, analytical protocol, what is actual analytical protocol? It's a very detailed text written in as a SOP, how to analyze and which solutions must be analyzed and what are the conditions of analysis. And also, as I mentioned, we have a capillary, which is packed in a capillary cassette, and this cassette is reserved for this certain application. If you work with the application at the beginning of the day, for example, for the determination of inorganic cations, you use a certain kit, certain solutions, certain conditions, and a certain capillary cassette. Then you wish to analyze inorganic anions in the same samples which you used before for the analysis of inorganic cations. You change the solutions from another kit, you take it from another kit, you change capillary cassette, and you start immediately analysis. The same you can do when you switch to another protocol for determination of organic acids, for example, or whatever. Every time when you switch from one protocol to another, what you need is to change the capillary cassette and solutions taken from another kit. That's all. This is the reason why it is so easy and fast to use many analytical protocols in one instrument.
Thank you very much. Our next question is, how many types of capillary tubes are used?
How many types of capillary?
Yes, capillary tubes.
Well, normally we use two types. It's a standard fused silica capillary with two diameters, inner diameter, 50 micrometer and 75 micrometers. These two capillary types are, let's say, standard, typical, and they used throughout all our analytical protocols. If we talk about the water analysis, this is absolutely enough. So this is 100% of our analytical protocols. They're used with either 50 micrometer inner diameter of the fused silica capillary or 75 micrometers. If we talk about the biotech analysis, pharmaceutical analysis, sometimes we use other types of capillaries, but for water industry, only this standard fused silica capillary.
Wonderful, thank you. Our next question. Are all of the tests for anion, cation, and organic acids and chlorination byproducts done using one instrument?
Excuse me, can you please repeat the question? I didn't hear it.
Of course I can. Are all of the tests for anions, cations, organic acids, and chlorination byproducts done using one instrument?
Yes, exactly. All analytical protocols, all examples which you have seen on this presentation were using one instrument, one Capel instrument. As I mentioned, I changed all our analysts. They changed just solutions and cassettes, capillary cassettes, when switching from one protocol to another. One instrument were used for all these protocols.
Okay, our next question is it possible to quantify hydrogen carbonates with capillary electrophoresis?
Unfortunately not. We see the peak of hydrogen carbonate. You have seen it also on all my slides, on all my electropherograms. But taking into consideration that hydrogen carbonate is very unstable in water and its concentration rapidly decreases because it goes out as a gas, it is impossible to accurately quantify it in the sample. So once again, we see the peak. We can say, yes, it is a hydrogen carbonate in the sample, but we cannot quantify it precisely.
And is the technique and the instrument approved by the EPA or any European testing bodies?
It is not necessary to approve instrument by the EPA. What is really important, to approve a certain analytical protocol. Our protocols were certified in different countries. In some countries, it is now even the national standards. We use the EPA protocol for the analysis of water for the determination of inorganic anions using our instrument, and it's enough. Additionally, our instruments have all necessary certificates like CE in Europe, for example, to be used throughout the globe without any problems. And of course, the instrument is being certified as a instrument to be used in analytical chemistry.
And how do run times compare with respect to ion chromatography techniques for non-organic ions?
The protocol which we offer is a very typical competitor of what is normally used by ion chromatography. It solves the same task, but faster, more accurate, and much, much cheaper. We separate inorganic anions one after another, exactly in the same way as ion chromatography does the job. But I mentioned many times in my slides, in comparison slides, that we can do it easier, faster, and sometimes more reliable, because sometimes you need to analyze your very complicated matrix water. It can be wastewater, for example, and you are not allowed to use the wastewater directly to inject in an ion chromatography. You need to use a certain first column. So there are two columns normally in case of fine chromatography and wastewater analysis. The first column, which let's say purify the wastewater, and the second column, which separate solutes. If there is some very poisoning things in the wastewater to be analyzed, you spoil the column, and thus you lose several hundreds or sometimes several thousands of dollars because the columns are very expensive. In case of capillary electrophoresis, even if you spoil the capillary, it is very hard to imagine, but even if you spoil the capillary by any components, you simply, in the very worst case, you simply take the capillary and throw it away and put a new one. Imagine capillary, 10 meters of capillary, which is definitely enough not only for you but also for your children, for all of your life. So 10 meters of capillary costs about $100. What you need is 50 centimeters capillary, which is almost nothing in terms of price compared with the fine chromatography columns. Thank you.
And Vadim, is it possible to check ozone with these instruments?
No, unfortunately not. It is impossible to check ozone with this instrument.
And in your presentation, you mentioned kits for analysis. Can you tell us what such a kit would contain and how much time you would need to start working with a new protocol using a new kit?
Yeah, okay. The kit contains, each kit contains two important parts. It is the text, the analytical protocol, let's say, where it is stated absolutely in all details how this should be analyzed, under which conditions this should be analyzed, and how the results must be proceeded. And also, kit contains the intermediate solutions, normally 2 to 3, sometimes maximum 4 small plastic bottles with prepared solutions. These solutions were prepared in Analytical Center of Lumex. In the standard operation protocol, in the analytical protocol, it is stated clearly how you must mix these solutions in order to get the final buffer. Normally it's just one procedure because these are already intermediate solutions. You make mixture, thus you get the final electrolyte. And also kit contains the calibration mixture number 1, means mixture with the highest concentration of the calibrated solutes. You can prepare the calibration levels, different calibration solutions by yourself, simply by diluting the initial calibration mixture, and thus you get all what is necessary to start immediately working with this certain methodology, with certain standard operation protocol. Once again, kits, each kit contains intermediate solution to mix to get the final buffer and the calibration mixture. Thank you.
And what are the chlorination byproducts that can be checked with these instruments?
I mentioned it at one slide that if you chlorinate, if you treat the water with sodium hypochlorite, you get normally chlorate, perchlorate, and chlorite. And these are the hazardous components, and all 3 components can be easily determined with our method which we offer and with our instrumentation. We have a kit to determine these 3 components in tap water after water chlorination process. Thank you.
Okay, so we have an audience member from a university lab who says that they need the instrument for method development. And they're asking if it's possible to run complex analyses with different stages or to provide multi-stage injections to get sample stacking.
Yeah, okay. Of course, all these things are possible because of the combination of the instrument features, and the features of the software. With the help of the instrument, you can apply simultaneously for the injection the high voltage and vacuum, the high voltage and pressure. These combinations gives you the possibility to play around with so-called sample stacking in order to enrich the concentration of your sample components and thus to get even better sensitivity than it was possible under the standard conditions without sample stacking. Besides, we have several protocols, for example, for the analysis of amino acids. When starting from a certain time point after the beginning of analysis, the instrument automatically applies the small pressure in order to facilitate the migration of slowly migrating components. So it means that we can pre-program different stages of analysis being included in one analysis. We can pre-program the changes of the wavelength, temperature, voltage, and so on and so on. So the unique combination of the instrumentation and software gives us the possibility to compose any conceivable schemes of analysis, including very, very complex schemes like very complex injection from different vials one after another, sample stacking injection, vacuum injection from the outlet end of the capillary, so-called injection from the short end to provide the very, very fast analysis time, and so on and so on. So it is really very, very flexible software and very powerful instrumental features. Thank you.